So today I'm going to talk about cytopathology diagnostic with multiplexed plasmonic biomarkers. For cancer screening, more and more pathologies promote cytopathology diagnostics when it's feasible. However, although it's widely used and recognized for some cancers, such as cervical cancer, because the tissue structure is lost, cytopathology samples may be difficult to interpret. To better understand the difficulty, let's take a look at the current procedure. First, there is the cell extraction through, for instance, fine needle aspiration, and there is a first assessment to determine whether or not there is a tumor and which antigens are going to be tested for further diagnostic. In order to do the analysis, pathologists prepare a paraffin block of cells or tissue that is then sliced and mounted on various microscope slides. Then they use staining to detect one antigen at a time for a given slide. The problem is that from one slide to the other, you cannot ensure that you are looking at the same cell. So you cannot really correlate between multiple antigens. And that explains why the diagnostics is quite difficult to make. And it's to overcome this problem that we want to propose an alternative using colorful nanoparticles to do the labeling of the antigens. And the individual nanoparticles will appear as distinct colorful dots in the samples, which will allow both multiplexing and quantitativity. So another advantage is that the labeling could be done in one single slide for multiple detection. And also the labeling would be done before fixation. Now that we have a bit of context, I'm going to explain better what are these plasmonic nanoparticles that we want to use and the various strategies for their visualization, after which I'll present a proof of concept for our technology on cells. So first of all, plasmonic nanoparticles are actually metallic nanoparticles, most of the time silver or gold, for instance. And due to their small size, they have unique optical properties and present a scattering peak, which will lead to a very distinct color. Another advantage of these nanoparticles is that they present no photobleaching and they present a really high scattering. So it's really easy to detect them. And we can detect single nanoparticles in a sample. In our case in particular, we have developed a chemical synthesis technique for alloy nanoparticles, so gold and silver. The advantage of these nanoparticles is that we can actually tune the color with changing the composition or the size of these nanoparticles. And we also have a patent on this synthesis. So we can, by using alloy nanoparticles of various sizes and composition, we are able to cover a wide range in the visible area. And we can also go to the infrared by adding some elongated nanorods to add more markers. Of course, nanoparticles by themselves are not specific in terms of binding. And antibody functionalization of nanoparticles through the classic EDC and HS chemistry ensures their specific attachment to a targeted antigen on the cells. So for instance, to show the specificity of the nanoparticle, we can compare the attachment of anti-CD44 nanoparticles on MDA-MB231 cells that are CD44 positive breast cancer cells. So here you can see on this image, the red dot that appear are all nanoparticles. And we can compare this attachment with pegylated nanoparticles that are actually non-binding nanoparticles. And you can see here that there are very, very few dots. Interestingly, we can also see that increasing the incubation time with the nanoparticles will lead to an increase of specific nanoparticles attachment, but the non-specific attachments from the MPEG nanoparticles will remain more or less the same. Of course, since we are talking about small nanoparticles, we have also to develop strategies in order to be able to see them. One thing that we need to remember is that the size of each individual nanoparticle is far beyond the resolution limit of an optical microscope. Here are, for instance, pictures of nanoparticles of 40 and 70 nanometer in diameter and also elongated nanorods, as are seen with a transmission electron microscope. It is important to understand that what we see under an optical microscope is not the nanoparticle itself, but the scattered light from the nanoparticle. So they will appear as small dots of different colors. So here, for instance, the blue one corresponds to the small nanoparticles, the red corresponds to the elongated nanorod, and the medium size is appearing in green. The most common way to visualize metallic nanoparticles is the standard dark field using a condenser. The role of the condenser is to collimate the incoming light and to ensure that the direct illumination will be deviated and will not enter inside the objective. When the light reaches the sample, it is partly scattered in an isotropic way by small scatterers. The light coming from the sample is then collected by the objective and can be detected. The image here shows, for instance, cells labeled with gold nanoparticles. However, in addition to conditions on the type of objective that can be used, the setup might be difficult to align. So because this system is rather complicated, it is not suitable for pathology labs and clinics. So that's why we developed a simplified dark field adapter in order to be able to see these nanoparticles on a simple pathology microscope for cells mounted on a microscope slide. And this is what we ended with. So here you can see a prototype of this adapter on a very simple standard microscope. We can compare here the image obtained with the standard dark field with the image that we obtained with the side illumination device. So the standard dark field shows a better contrast for the cells, but as you can see in this specific area, on the right image, you can see some small blue nanoparticles that are not so visible on the left side. So now that we have a bit more of context and understanding of the technology, we can move to the very point of all this, the application in cytopathology. To demonstrate our technology, we compared the performance of our technique to existing labeling procedure, such as immunofluorescence. So here on the left, you see immunofluorescence performed on MDA-MB31 cells with anti-CD44 fluorescent label. So the red coloration on the membrane shows the presence of the CD44. And we did the same with the functionalized nanoparticles. And you can see here the dots that appear on the membrane. And there is also a phalloidin stain to show that there is a good correlation between the nanoparticles location and the membrane location. Then we also did the demonstration on two different cell lines expressing different levels of CD44, EGFR, and KB1.1. So the immunofluorescence that you can see here on the top showed the expression of the three markers on the MDA-MB231, while the cell lines 661 and W is positive for the KB1.1, while it's negative for the two other markers. So we then compared these immunofluorescence results with the immunonanoplasmonic labeling. And we could see a quite good agreement. So we used three different types of nanoparticles to assess the presence of the three different markers. And we found quite agreement with the previous methods. So as a conclusion, I'd like to say that we have promising results for a new immunolabeling protocol. We have a very good control of the alloy nanoparticle synthesis and improved optical detection and the good agreement with immunofluorescence technique. We are now working on hyperspectral imaging strategies to increase the number of markers that we can detect. Also, we are working on automated image processing and classification, digital pathology, and the application of the technique to the PD-L1 detection in lung cancer. I'd like to thank all the lab mates and collaborators that have contributed to this work and invite you to have a look on our publications if you want to have more details. And I'd like to thank you for your attention.
Cécile, could you speak more about how multiplexing at the single cell level would be an improvement compared to current procedures?
Yeah, sure. Thank you for the question. So as I explained before, most of the time pathologists now can do multiplexing, but only on several slides. And because the cytology, in cytopathology, the structure of the tissue is lost, so we cannot ensure that we are looking at the same type of cell from one slide to the other. So that's why, and in some application, I'm thinking in lung cancer and PD-L1 detection, for instance, in non-small cell lung cancer, PD-L1 can be expressed by various types of cells, but the impact of this expression will not be the same on whether or not the patient is eligible for some treatment. So that's why it's really important to identify both the type of cell and the marker that we're interested in.
Excellent. Thank you. And I saw Dr. Diana Royce's hand is up. So Marshall, if you want to unmute her, that'd be great.
Okay, Cécile, thank you so much for a very interesting and impressive overview of your project. In terms of your own personal development, what did you find in this project to be the most challenging component, but also the offering you the most scientific growth as a researcher and as someone contributing to innovation in the field of nanomedicines?
Wow, that's a big question, and I thank you for that. In my as you can see, this project is really, really huge, and a lot of persons are involved in this, from the nanoparticle synthesis to the nanoparticle functionalization, and also there is a huge work that has been done on the nanoparticle visualization itself. So for me, I think that one of the biggest challenge of this project is precisely the fact that so many different expertise are required to reach the final goal. And I've been mostly working on the imaging part, and it's very interesting to see that I mean the whole project grows, all the parts grow together. So if we have some new, for instance, new type of nanoparticles that are used, and then we need to go back to the imaging system and ensure that everything will still work together. So I think that's the most challenging part, probably. And also we have inputs from the pathologists that say, for instance, oh, we want also to be able to see counterstaining, and counterstaining will add a really strong, colorful background. So we also need to work on that, for instance.
Well, thank you for that response. I think this project is an example of a terrific opportunity to prepare yourself for industry opportunities, because the things that they're looking for are ability to work across disciplines and across areas of expertise and teamwork, and seeing how all those small pieces fit into a bigger picture. So congratulations, and thank you so much for this excellent presentation.
Thank you.